It was good today. Got heredity. Did I spell that wrong? Yeah, I did. Okay. What's the definition of heredity? Heredity is the passing of traits from generation to generation, from parents to offspring. What are genes? Genes are specific, like, bro, this iPad's not staying. Bro, my bad. Wait, where's my phone? What are genes? I already said that. What are genes? Genes are they're like specific sequences of DNA that code for a specific trait. Alleles are versions of a trait, or like just like the version, like, blue or green, like they're just versions of the same gene. So yeah, what is locus? That's like the specific physical location that you'll find a gene on a chromosome. Categories of chromosomes in humans. Categories of chromosomes in humans. What does that even mean? Probably like diploid or haploid, maybe again. So categories of human chromosomes in humans. So we got diploid, that's like the normal number of chromosomes in a cell. So that's 46 in humans. So then haploid would be 23. So define diploid. Diploid is like the normal number of cells. So that's going to be like in all somatic cells. And then gametes are going to have haploid cells, haploid number of chromosomes. I feel like I'm missing something for that, because why would I ask? I don't know. So what are the 3 cell types? There's 3 cell types are germ cells, which are diploid, which lead to gamete cells. They undergo meiosis. So the germ cells undergo meiosis to form gametes, and gametes are haploid, while germ cells are diploid. And then there's like the regular somatic cells, which are just diploid. So what I meant by I already talked about classifications. Mutation. So if you have a mutation to a somatic cell, it's not going to be shown in the offspring. But if you have a mutation to the gamete or the germ cell, then you're going to have that shown in the offspring. So relation to your parents, you're 50% related to your parents and about 50% related to your siblings. But it's not like set 50% to siblings, because like independent assortment, other variations, stuff like that could cause it to be different. So compare genotype versus phenotype. So genotype is just the genetic expression. How you have like deja vu sometimes? It's like you just like have a feeling that you did this, but like I swear I have not done this before. Like compare genotype and phenotype. I feel like I had a dream about this like 3 months ago, and then like earlier, like 2 days ago, I had like the deja vu feeling of like when I was planning out what I had to do to study. Like I had that feeling, but I never opened like that crusher before, right? But like I just had that feeling. But anyways, so phenotype is physical expression of genes. Genotype is the genetic expression of the race. Like, do you have dominant, or are you like heterozygous recessive, heterozygous dominant? Homozygous dominant, homozygous recessive, or are you heterozygous? That's like genotype. Phenotype is just what you physically see. So dominant versus recessive allele. So the dominant allele will always be shown. Recessive allele will only be shown if you have 2 recessive alleles. So if you're homozygous recessive, it will only be shown. But in other cases, like if you're heterozygous or homozygous dominant, then the dominant will be shown. So dominant will just like take over the recessive. So 3 allele combinations. So you got homozygous, which is the same allele. So homozygous recessive, homozygous dominant. You got heterozygous, which is 1 dominant, 1 recessive. And then you got, I literally just looked at this, but it's like hemozygous or something. Hezizygous. Hezero, hemizygous. Damn, I was looking close. Hemizygous. Hemizygous is just when one of the alleles is present and the other one is just not there. So it's kind of like the XY chromosome, because the Y chromosome is shorter. It's not going to have some of the alleles for that, like gene, for like that copy. It's not going to have like the exact same copies of DNA than the X chromosome. So yeah. So what are the 3 laws of inheritance? So first of all, there's this law of segregation, which says basically that the gametes will include only like 1 set of chromosomes. So when they combine, it'll become like diploid. So That's kind of just like describing the process of meiosis. Second thing is independent assortment. So it's like when during anaphase, I mean during wait hold on during metaphase one, when the homogeneous chromosomes align at the metaphysial plate. Damn, I feel like I low-key know like a decent amount of stuff now. Like, well, it's like low-key kind of like it's kind of nice, kind of like it's like I'm starting to understand some of like the terminology that other people use, like when they start talking about more advanced biology. I'm like, wait a minute, like I understand that, and it just makes me feel like an unk. But that's all. I don't know. I just want to say that. So the second law is independent assortment. So it's like when they meet up at the metaphysial plate, you don't know which chromosome will be pulled to which cell, because they could be like just on either side of it, so it doesn't matter which one is which. Did that make sense? It's like when they line up, you don't know which gene will go to the other, and they're independent of each other because one gene will not, like one chromosome will not affect how the other chromosome is, so that's why it's independent. The third one is what is the third one? Wait, how do I not remember this? Wait, let me just think, bro. There's independent assortment, then and then there's the like dominant or recessive. You'll have a dominant allele, you have a recessive allele. That's kind of common sense for that one. So what is experimental heredity used for? It's simply used for like determining the genotype of either parent. It's just like what is the genetic information? Like what is the, like how is it like, well, I don't really know what to say. It's just like just to find out what the genotype is. Let me just see. Used to determine the genotypes of individuals. So what is a true breeding cross? A true breeding cross is when you mix homozygous recessive and homozygous dominant, and then in that case you're going to get monohybrids because all of the offspring will be heterozygous, so they're going to have one of each gene. Terminology for different. Bro, I'm so mad about OChem, bro. I'm so mad about OChem and Chem 102, bro. Those could have been easy 4.0s if I just didn't get screwed by the lab, bro. Makes you so sad sometimes. Anyways, what was that? Terminology for different stages of experimental heredity. So the first is going to be the parental, then it'll be F1, which is the filial, like the first filial hybrid, and then you're going to have second filial, F2 generation. I don't really know why I asked that, but anyways, what are monohybrids? Monohybrids are just, they're just heterozygous. They have one of each gene in their genotype. Genotype and phenotype ratio of the last generation breeds. So when you breed the monohybrids, you're going to get a genotype ratio of 1 to 2 to 1 and a phenotype ratio of 3 to 1. It's just if you use a Punnett scale, it makes sense. What are test crosses used for? So what parents are used? You're going to use that unknown and you're going to combine it with a homozygous recessive because you're going to know that it's a homozygous recessive because it'll have that recessive trait. And then with that, you could determine what the other parent is. So it's just used to determine the genotype of an unknown. So it's kind of like experimental. It's like, it just makes sense. What are the two types of Mendel crosses? You're going to have monohybrid crosses, which is just talking about like one gene. So you can have like capital B, little b, just talking about like eye color. And then you have dihybrid crosses, which is going to be like big B, little b, big D, little d. That was like, wait, pause. But it's like, we could say like big A, little a will control like hair color, and then you could have big B, little b, which will control like height or something like that. So that's dihybrid cross. What is a dihybrid cross? Is when you're crossing two different genes. The phenotypic ratio will be 9 to 3 to 3 to 1. Wait, 3 plus 3 is 6. Yeah, so 16 total. It would be a 9 to 3 to 3 to 1. Yeah, phenotypical ratio. What did this dihybrid cross confirm? It confirms independent assortment because from that phenotypical ratio, Instead of it being a 3-to-1, that would, like, if it was 3-to-1 ratio, then that would confirm like, yo, it's dependent assortment. That means like wherever one gene goes, it'll depend on like where the other one goes. But because we're getting a 9-to-3 to 3-to-1, that shows like that the genes assort independently of each other. So yeah. What is the difference between codominance and incomplete dominance, cause and effects? So incomplete dominance is when you have two like, they're not like fully dominant, but like, you know, they're incomplete dominant. So like it'll form a blend of colors. You'll get a third phenotype. It'll just be a blend. So if you have like blue and you have white, then if they combine, they're gonna, I mean, if you have like red and white, you're gonna get like another pink one. You know what I'm saying? So that's gonna be like your third phenotype. What is codominance? Codominance is when you have like two like fully dominant alleles, and then they'll both be shown, like they're both like equally dominant and they're both be expressed in the phenotype. So if you, it's like going to be like, if you have red and white, it'll be like red and white spots. So just to clarify again, incomplete dominance is when you have two dominant alleles, like two or more, I guess, like you have one or more dominant alleles and they'll be expressed in blending, and then codominance, they'll both be expressed but they won't be mixed. That 3 is just, I don't know. So what is epistasis plus example? So epistasis, is that how you say it? It's when one gene will control the expression of the other. So like the phenotypic expression. So it's like baldness, right? If you have the baldness gene, it doesn't matter what kind of hair gene you have, you won't have hair. So that's kind of like what it means. Like it'll dictate the expression of another gene. What are multiple alleles? So you could have like multiple alleles, you know, like more than two. So it could have like blue eye color, red eye color, brown eye color. So yeah. What is pleiotropy? So that's when a single gene will control multiple traits. An example of this is like in the plant cell, something will control the color, the height. I don't fully remember the example, but it's like, yeah, so a single gene will control multiple traits, phenotypic traits, so plant height, plant color, and plant texture. Those are the 3. Polygenic inheritance is kind of like the opposite of pleiotropy. Polygenic means that multiple genes will contribute to a single phenotypic trait, something like height or weight. Those are just 2 examples. So sex-linked versus autosomal linked genes. So sex-linked are just genes that are on the sex chromosomes, like the X or Y chromosomes. Autosomal linked genes are just regular genes. So like sex-linked genes, they'll just be passed on via the sex chromosomes. Autosomal is just all the other chromosomes. What is a carrier? Can men be carriers? So a carrier is someone who has the condition, like allele, like let's say like a bad allele, but they don't express it phenotypically. So in that case, that would be like being heterozygous, right? However, for men in sex-linked genes, men only have one X chromosome. So if they get that recessive chromosome, they're not going to just be a carrier. You know, they're going to be expressing it because they only have one X chromosome. They're not going to have a dominant allele to match that recessive one. So men cannot be carriers for sex-linked chromosomes or genes. I mean, if only a dad had an X-linked chromosome, can the sons have that condition? No, the son cannot have those conditions because the dad will only provide the Y chromosome to the sons. The X chromosomes will be given from the mother. So a dad cannot give an X-linked condition because the dad's only responsible for giving the Y chromosome. What is the difference between sex influence and sex-linked genes? So sex influence is just kind of based on gender, like short hair or long hair, while sex-linked is just on the chromosome, you know? That's like the main difference, I guess. Something I wanted to say for polygenic inheritance, it'll usually be like a range. So you could have like super obese people, like heavy people, or things like that. Sex-linked sex influence. So expression is influenced by sex of individual carrying trait. Example, baldness. Oh, okay. So if even if a so if baldness is a dominant gene, and even if a female has it, it won't matter because she will not go bald because she's female. I think. Just as expression influenced by sex of individual carrying trait. So like a heterozygous male will become bald and a heterozygous female will not become bald. Interesting. What is the purpose of genomic imprinting? So genomic imprinting is just when you methylate DNA, because you have 2 copies of each gene in each cell, so you don't want to be reading 2 at the same time. So the body will just deactivate them by methylating them. What is penetrance? And then what is expressivity? So penetrance is like if a gene will be expressed. So you could have complete penetrance or incomplete penetrance. So like if you have complete penetrance, then wait, hold on. If you have complete penetrance, then 100% of the individuals that carry that affected gene will be blind. So penetrance is the probability that an organism with a specific genotype will express the corresponding phenotype. Okay, so penetrance is a probability. So then there's complete penetrance, which is when 100% will show, and then incomplete penetrance, which is when some will show, like not all of them. And this is talking about like who have the affected alleles. And then expressivity is like how much you express it. So it's like body hair. Like you could have high expressivity of body hair, you could have a lot of body hair, or you could have low expressivity, which is like a little body hair. It's just like a range of values, like, so the variation. So the key difference, what I would say is like penetrance is, are you there or are you not? And then expressivity is like the range, like in the middle. What are the 5 situations of penetrance and expressivity? So first you could have complete penetrance, which is when everyone will have that, everyone will show that thing. Then you could have incomplete penetrance, which is when some of them will have it. You could also have, like, okay, so I said incomplete penetrance, right? You could have variable expressivity, which is when some will show more, some will show less. You could also have variable penetrance and variable expressivity, which is where some of the people will not have it at all, some people will fully have it, and some people will just be in a range. So you never know, like, what I'm saying? And then you have unexpressed, which is like no one will show it. So, damn, bro, Loki, boy, this is a long chapter, bro. Not chapter, I mean topic. So, what is X inactivation? I meant, what does it make? It makes a bar body. How does it methylate the X chromosome? And what decides what is on and off? It's completely random. So X inactivation is, how there's going to be 2 X chromosomes. We don't want 2 X chromosomes, so we're going to deactivate one of them, make it into a bar body by methylating it, and it'll just be inactive. So that leads to the question, why is Turner syndrome a bad thing? Because Turner syndrome is when you don't have an X chromosome, like you're missing one. Well, on the bar body, not all of the chromosomes are actually inactivated. So some of them are still expressed, so you need to have it still. What is heterozygous advantage? Is just when having both of the alleles will help, both of the genes, but like both of the alleles will help. An example is like sickle cell anemia. If you have sickle cells, then you're not susceptible to malaria. But if you have sickle cells, then it's harder for blood. Like blood does not stick to like the red blood cell, and it's not the oxygen. It's just the red blood cells will just die quickly. So, and if you have regular blood cells, like you're carrying oxygen, like they're healthy, but you're susceptible to malaria. So if you have heterozygous, if you have both of them, then you're basically like you got the best of both worlds. What are linked genes? So linked genes are just genes that are close together, and they have like a higher frequency. Like the term is called recombinant frequency. It's just a higher chance of them being like crossed over to like a different, like you know, like doing crossing over, doing like synapses, like when they form the tetrad. I'm just throwing out terms, but it's like when you have that, like the recombinant frequency is the chance that those two genes will move to another thing together. The highest you can have is 50% on the same chromosome. Why? Well, if you're so far apart, then there's a high chance of you being replaced, but then there's also another chance that you also get replaced on the other same thing. So it's just like you will end up on the same thing maybe in the end. Like it kind of, like I don't really fully get that part, but it's like I think that's just an easy memorization thing. So yeah, I just want, like, my dad explained linked genes properly. So yeah, during meiosis 1, they cross over. Closer the genes are on the chromosome, the less likely they are to be separated. So yeah, linked genes are just closer apart. They have low recombinant frequency because they're close apart. Yeah, wait, hold on. Now I'm looking. Yeah, so the further apart, the higher recombinant. So okay, so linked genes. So two genes are linked genes if they're close together, but then they have a low recombinant frequency. Okay. What do pedigrees show? They just show like male or female, or like how different traits get passed on through families, kind of. What are the four inheritance pattern categories? So you could have, first you could have recessive or dominant, and then you could have X-linked or autosomal. Bro, pedigrees were low-key making me lose my brain, but I can't tell between autosomal or X-linked, but the only way I can tell is like, yo, if more females have it or more males have it. But it's kind of hard to like say, like, based on the other thing, like if the dad is unaffected, then he must have an affected, he must have an, if the dad is, like, I don't get that part, bro. I need to like explain it more. So what are genetic defects there? What are genetic defects? Genetic defects are just, what are they caused by? One of them was caused by many other factors. The other was pedigrees, yada yada yada. Caused by mutations, environmental factors, or other damage to chromosomes. 4 main genetic defects. One is point mutation, which is just mutation on the gene. Second is damn, that's loud. But I should know these, bro. There's traversion. So okay, wait. There's point mutation. There's transversion, traversion. Wait, traversion. Okay, wait. Wait. I'm getting very confused. Okay, so the 4 genetic defects, 4 main genetic defects. First one is point mutation. Second is non-disjunction. Third is chromosomal aberrage. And I don't remember the last one. Chromosome breakage. Okay, that was rough. That was very rough indeed. 5 points of, what are the 5 types of point mutation? So first there's like frameshift mutation, which is caused by like insertion or deletion. Then there's transversion, transition, frameshift, and then forward, and I'm assuming the last one would just be backwards. Right? Okay, yeah. Okay, let me do that again. So the four main genetic defects. Four main genetic defects. So first we have point mutation, we have chromosome aberration, chromosome breakage, and then we have non-disjunction. 5 types of point mutation are frameshift mutation. Damn, this kid's doing a wheelie? We have frameshift mutation, we have translation, no, yeah, translation. No, not translation, transition. Why is this doing, why am I, why can't, I need to focus, okay. So frameshift, transition, transversion, forward and backwards. So what does each one imply? So frameshift is when you insert or delete a nucleotide. That'll cause the entire code to be shifted to the right or to the left. So that'll cause like the codons to be messed up. Like you could have a stop codon earlier and it'll make it like a nonsense mutation or something like that. Yeah, okay, so now what's the other one? Transition. Transition is when you switch a purine to another purine. So it's like if you go from A to G, while transversion is when you switch from purine to pyrimidine. So you go from adenine to thymine. Yeah, that was low-key rough, bro. Transition is from adenine to guanine, so between purines, while transversion is pyrimidine to purine. Okay, what is forward? Forward is when you go from like the normal, like the wild type allele to a mutated version of it, and then backwards is when you go from the mutated back to the wild type or the normal type. What is mosaicism? So that is caused when, so during non-disjunction, perhaps I should explain the four main genetic defects as well. So, oh man, okay, so we went to the point mutation, then you have non-disjunction. Non-disjunction is when like an incorrect number of like genetic material, like chromosomes, move from the cell, like are like they're not divided equally between the cells. So that'll cause mosaicism. And is that how you even say it? Mosaicism. That's like when you have like more DNA or more chromosome, and then it'll cause that cell to be genetically unique to the other cells, and then it could replicate and spread, and it will cause issues in the body. Yeah, so non-disjunction is when one or more chromosome pairs and chromatids fail to separate. Could happen in mitosis or meiosis. So mosaicism occurs in the cells that undergo non-disjunction. So then some of the body cells are going to have extra or missing chromosomes, and that'll not be good. So it can occur during like when the embryo is being developed. So like if one of the sperm cells or the egg cells had an issue or like went through non-disjunction, then that could cause it. But it also can happen from nucleotide mutations, because like, it'll just cause an issue, I guess. What is trisomy 21? You have an extra chromosome 21, that leads to Down syndrome. What is aneuploidy? That is just the general term used for like when you have too much chromosome or too little chromosome. Like it's just like incorrect amount of chromosomes, like if you have extra or missing. What happens to non-disjunction in meiosis 1 and meiosis 2? This is a good question. So meiosis 1, the issue will happen during when the homozygous chromosomes are split. So you're going to have more chromosomes on the left than the right, or the other way around. So the left will have more, the right will have less. So then when you go into divide further in meiosis 2, you're going to have, aren't all of them going to be messed up? Yeah, all of the cells will be messed up. All of the sperm cells or all of the get will just be messed up. Some will have, two of them will have more, two of them will have less, because when you split it from meiosis one, like splitting the homozygous pairs, one side will have less, the other side will have more. So then when you split it, the two will have more and the two will have less. Now on the right hand side, if you have meiosis two, then anaphase will split the sister chromatids, but anaphase one will have gone well. So you're gonna have one normal cell and another normal cell. So then when you divide them further, you're gonna have two messed up cells, one with more, one with less, and then two normal cells. Nice. Okay. Damn, that was looking kind of tough. Okay. What does aneuploidy mean? I already talked about that. Okay, what does karyotyping do? It's just simply a way to see all the chromosomes. Happens during metaphase, because that's when all the chromosomes are like lined up and condensed. What are the 5 types of chromosomal aberrations? So you could have inversion, you could have single replacement, you could have double replacement. It's like the chemical reactions. Okay, wait, it's not replacement, but so you can have inversions, you could have duplications, where you'll, yes, so, okay, wait, let me restart. So you can have duplications, you can have inversions, you can have one-way translocations. So that's the other one I was confused about. Two-way translocation and then deletion. So duplication is when you just copy the same genetic material onto the other one, and you could tell it by its, like, if it's longer or not. One-way translocation is when you take the section of the chromosome from one and just put it onto the other one. That's one way. Then two-way is just both of them switching, flip-flopping. Inversion is when you take one piece and then you flip it and then you stick it onto the other one. Deletion is just when you delete a part and it's just gone. Yeah. Okay, so what are 2 cells to suppress cancer? Well, let me just make sure I have everything. Inversion, one-way translocation, two-way translocation. Tell me what you say now. Okay, yeah. Chromosomal breakage is when it breaks. So it's kind of similar to deletion then? Yeah, it just says deletions or rearrangement. So chromosomal breakage kind of leads to the different types of aberrations, and like one-way translocation, two-way translocation, etc. So what are 2 cells that suppress cancer? One is proto-oncogene. Yes, proto-oncogene. And the other is tumor-suppressing genes or cells. Proto-oncogene cell gene, I don't really know. So what does proto-oncogene do, you may ask? Well, it regulates regular cell growth. It tells the cell to grow. Now, if that cell gets mutated, then the cell will keep on growing and it'll get messed up. Now, okay, so it says if mutated, it becomes oncogene, which causes cancer. An example is the RAS gene, RAS kids, okay. Bro, there's literally a lot of information to remember for this DAT, but I'm just clocking it, like I'm not just clocking it, like I knew it, but it's like, damn, it's like a lot, bro. And the other one is tumor suppressing genes, which is like, it'll tell the cell to stop growing, like it checks the checkpoints, and it'll prevent the cell from dividing too much. And yeah, so if it's like mutated or messed up, then the cell will just keep on dividing abnormally, and it'll cause tumors and cancer. So an example of this one I remember is the p53 gene. This is something we learned about earlier in an earlier thing, like during cell cycle, I think, right, bro? I don't even know. Damn, bro, I need to look into that. But yeah, so how do viruses cause cancer? They'll just cause mutations to those 2 genes that suppress cancer, and they'll just cause issues. Like viruses, they'll bind to the cell, and then they'll like combine their DNA with the hosts, and then everything will just get messed up. What are mutagenic agents? So those are just things that cause mutations. Or I thought they were just the same things like carcinogens, but maybe not. There's like radiation, like UV or X-rays. There's also chemicals, which is like mustard gas or cigarettes. And then there's infective, which is like bacteria, I think, or I don't really know. What does it even say here? Helicobacter pylori, human papillomavirus. Yeah. Yeah, bro, we're not going through those examples because I genuinely don't remember those ones. Um, what is an extranuclear inheritance? What is that actually? Um, so it's like, so extranuclear is like, it's outside of the nucleus. So you might be asking, like, what other genes are outside of the nucleus? Well, I'm glad you asked. Mitochondrial DNA. Mitochondria contain their own DNA, and that mtDNA that comes from your mom. So you can inherit that from your mom. That's basically what it's talking about. I'm pretty sure. Let me see. Yeah, genes also exist outside the nucleus. So defects in the mitochondrial DNA can reduce cells ATP, and that comes from the mother, because the mother gives the mitochondrial DNA to the egg. So it's just caused by that. So all mitochondrial related diseases are maternally inherited. What are lethal genes? So some genes will just cause death. So let's say if you have two dominant, then you die. Then that'll affect the phenotypical. That'll affect the phenotype ratio. You know, if one of them is dead, then it's not going to work, right? So if you have like heterozygous, like a monohybrid cross, and the dominant is dead, then you're only going to have a 2-to-1 ratio. You know what I'm saying? So it'll just change. So, yeah. Or it works the other way. If you have homozygous recessive being the lethal, then you'll have a 1-to-2, because the third one will be dead. So, yeah. That's basically all. See you tomorrow.